Hi class, it's Labria and I'm going to be talking to you about HIV and AIDS. So a little bit about the disease. HIV infection is a primary immunodeficiency disease caused by the human immunodeficiency virus. So there are two kinds. HIV-1, the point of origin is Central Africa where it's more severe. HIV-2, the point of origin is West Africa, which is less severe. And as far as prognosis goes, HIV, if untreated, can lead to AIDS or acquired immunodeficiency syndrome. There is no cure, but with medication and lifestyle changes, you can reduce the progression of the disease. HIV originated from a chimpanzee in Central Africa, and it may have been transmitted to humans in the late 1800s. It existed in the United States since at least the mid to late 1970s. A little bit about the etiology. HIV is an infectious disease caused by the human immunodeficiency virus, as stated before. It spreads via sexual contact, blood from blood transfusions, but this risk is small because they do test the blood that they use before they do these blood transfusions. Sharing needles from the contaminated IV drug paraphernalia. And it also spreads from mother to child during pregnancy, childbirth, or even breastfeeding. So when HIV turns into AIDS, there is an increased destruction of CD4 plus cells, which are the white blood cells. And you can have an infection for years before it turns into AIDS. AIDS is diagnosed when the CD4 plus T cell count falls below 200. It's also important to note that HIV does not spread through hugging, kissing, dancing, or shaking hands with an infected individual. I think that's very important because a lot of times there's stigma, that you may know that somebody has HIV and then people, they don't want to be around them. They think just being close to them can cause them to get HIV, which is not true. So a little bit about the pathogenesis, what's happening inside your body and the cycle that this is in which is happening. So we start with the infection of mucosal tissues, and then there's death of mucosal memory CD4 plus T cells, which again are the white blood cells. Then there's infection established in the lymphoid tissues, the lymph nodes. And then it spreads throughout the entire body. And then we have establishment of chronic infection that leads to the increased viral replication. And then finally, that's when AIDS occurs and there's destruction of the lymphoid tissue and depletion of the CD4 plus T cells, as mentioned before. So I included some pictures here to help with our visual learners because I am indeed a visual learner. But this is a picture of an HIV particle here. And then here is a picture showing early HIV infection. So you have the virus here that binds to a CD4 receptor, and also the co-receptor helps with this process. So then it binds to the receptor and the co-receptor here, which is these red squiggly lines, and that then allows the virus to fuse with the host cell, and the DNA and RNA then gets injected into the host cell here. So another visual representation. So you have the HIV virus. It fuses with the cell, and this is the CD4 plus cell, which is the white blood cell. So then you have the viral RNA that gets injected after the fusion happens. Then you have, and then it migrates to the nucleus where you have reverse transcriptase that transforms into DNA. Then that produces the messenger RNA, and then you have the viral protein here. And eventually that comes out of the cell, and that's when the HIV virus is replicated, and then it goes on to another host cell to infect another host cell. So here are some symptoms of the disease. You have night sweats, mouth ulcers, fatigue, chills, muscle aches, rash, fever, and swollen lymph nodes. So here are some risk factors that I thought was also important to mention. Unprotected sex. If you have a STI, a sexual transmitted infection, and this is because a lot of these sexual transmitted infection leaves open sores and pores in which it's a little easier for the virus to enter into the body, and use of IV drugs. So next we have prevalence, and we'll start with in the Saharan Africa. So about 24.8 million people are affected in the Saharan Africa. And in Latin America, 1.7 million people are affected. And I'll skip over to Eastern Europe, 1.5 million people are affected. And in Central Asia, 5 million people are affected. And then in the United States, it's less people affected, more than 1.2 million people. And that is because in the United States and other industrialized countries where access to medication, care, and prevention is much greater, and the number of patients diagnosed with and dying from AIDS is stable or declining. That's why it's noted that in the United States, it's a little less compared to the rest of the world. Also, other countries have more limited access to testing and medication for treatment, and they have limited information for prevention because of the effects of gender inequity and harmful social norms that drive the transmission. So some other key numbers and demographics, and all of these numbers came from the book, so there may be some slight differences when you try to Google the numbers, but these numbers came from the book. So 36.9 million people are living with HIV worldwide as of 2014. 1.2 million AIDS deaths. There are 34.3 million adults, and of that number, 17.4 million are women and 2.6 million are children who are less than 15 years old. So as far as race and ethnicity goes, racial and ethnic minorities and women of color are more affected. 41% are African Americans, especially in the young age group, and 20% are Hispanics. In the Northeast and South report the highest numbers of HIV and AIDS diagnoses. The South reports about 45% of new AIDS diagnoses. Northeast reports 24%, the West reports 19%, and the Midwest reports 13%. So, how's it diagnosed? There are several different ways that HIV can be diagnosed. So, the CDC recommends that HIV testing be a part of your routine care for all adolescents and adults. I know for females, when you go to get your annual pap smear, they do often ask if you want an HIV test while you're there, and it should be free of charge. So I know that fact. And as far as the FDA, there are approved various methods, including the rapid HIV assays that use blood, serum, or oral secretions with results in little to 20 to 30 minutes. So also with that, it's important to know that the positive results must be confirmed by further lab testing because you can get false negative tests that happen in the initial period of the HIV infection. That's why it's important to get multiple tests and confirmation with this rapid test. And you also have serum and plasma specimens tested in approved labs using the HIV-1/2 antigen antibody combination immunoassay. This other test here is called the ELISA, enzyme-linked immunosorbent assay, and this is used to detect the HIV antibodies. And when this test is positive, the Western blot test is used to confirm the presence of HIV antibodies. So again, there's a lot of like initial testing and then another test to confirm that initial testing. And then for neonates, the best method is to culture the virus from blood in the peripheral tissues. So here are some other ways to test and monitor the progression of the disease. There are a lot here. So you have your HIV antibody. You have your complete cell count with differential and platelets, CD4 plus count percentage, quantitative plasma HIV RNA, resistance testing, quantitative immunoglobulin, chemistry panel, lipid panel, energy panel, gonorrhea, chlamydia, and trichomoniasis testing. And like I said, this is gonorrhea and chlamydia testing. It can be done because a lot of STIs occur with HIV a lot of the time. And then you have renal or abdominal ultrasound, urinalysis with urine protein, and creatine, hepatitis A, B, and C serology, blood cultures, TB screening, chest radiograph, HLA-B5-SO1 testing, tropism testing, Papillary test, pregnancy test, serum VDRL, dilated retinal exam, and CMV antibody. So yes, that's a lot of tests used to diagnose and to monitor the progression, which is good. And then as far as treatment, there are antiretroviral therapy recommendations. This is the use of multiple medications or the heart that involves administration of multiple antiretroviral agents. And you also have your NRTIs, your nucleotide RTIs, NNRTIs, protease inhibitors, fusion inhibitor, CCR5 antagonists, integrase transfer inhibitor, and there are other treatments and vaccines. You got your antibiotics, antivirals, antifungals, antiparasitics, and antimycobacterial medications. And then you have vaccine conferred active immunity. But with all that being said, there's also noted that there's not an official vaccine to help prevent HIV. They haven't developed that yet. But there have been different trials of vaccines. None of the vaccine trials have been successful, and several have been stopped in phase 2. And at this time, like I said, there's no effective vaccine available for the prevention of HIV. And I also like to point out that some of the antivirals that maybe we may be more familiar with because of a lot of the commercials now that are out are the Biktarvy and Dovato. Those are some common antiviral HIV medications that are out right now. Here are my references, and thank you. Please ask any questions, and I will do my best to answer the questions as thoroughly as possible. Thank you for listening, and I can't wait to hear from you all.